Okay, we are back again with the blood vessels. In earlier few lectures, we have seen histology of artery, different types of arteries. Now, in relation with the blood vessels, we will see here today that is another blood vessels, that is veins. And veins, the definition of a vein is the blood vessels which bring back blood to the heart, that is vein. So here, like that, artery veins are main vein which originate from the heart, which bifurcate into different branches of veins, and the smallest branch of a vein which leads into a capillary, that is called as a venule. So here in this diagram you can see a vein collecting blood from the capillaries. That capillary end into a venules, and veins end into the vein, and like that this blood flows from arterioles to large arteries to arterioles, then in a capillary network, then again that blood unite, all capillary unite to form a small venules unite to form the veins. So today in this lecture we will see mammalian histology, a detailed histological structure of these venules and vein. Now, as we have seen histology structure of artery, there are three layers we seen. The same three layers are present in a veins also. The difference is the internal lumen, that is hollow cavity inside the blood vessels of a veins. This is quite larger as compared to the arteries. Here the same three basic layers you will see as a tunica adventitia, that is outermost layer, tunica media and tunica intima. But that elastic and muscular components are very less prominently seen, and you cannot even distinguish these three layers in a veins because there is no limiting membrane or layer present in between these three, tunica adventitia, media and intima. Again, that smooth muscles layers which is used for causing the dilation and constrictions of veins, they are present, and that is the reason why veins can accommodate large amount of blood volume. First we'll see here the structure of venules, that is they have a clear tunica intima layer without any elastic fibers, and tunica media with one or two layers of muscle fibers, and tunica adventitia which is fused with the surrounding tissue. So the structure of arteriole and venule is much more similar. Not much difference is there in case of venules and arterioles. Now here in this color photograph of a slide under microscope, they are showing a smallest vein. Now it look like this in a section. If you take a section of any major organ in a body and stain it with eosine and hematoxylin, and when you observe that section under microscope, you will find such many structures. Such many structure you will see. Sometimes they are hollow vacuoles, or sometimes they are with this such kind of a red beads. All these red beads indicate the RBCs. So because of this particular pattern of red beads in a hollow cavity, you can identify that yes, it is a some kind of a blood vessels. Thereafter, when you go in a detail of this wall structure, you come to know whether it is a artery or vein. So here you will see clear structure of a peripheral cell layer, that is endothelial cell layer, with distinct blackish color nucleus, and after that there is no any such distinct remarkable layer you will see around these blood vessels. So that way you can come across that this is a venules, a vein, a large size lumen, and a surrounding endothelial cells indicate that this is a venules. Again, in this photograph of slide sections, this is a vein, a very large and not a very precisely round but little bit collapsed structure that also indicate that this is a vein. If it is arteries, then it must be very precisely round structure because of that muscles and elastic fibers layer, tunica adventitia and tunica media. These arteries remain very much rounded in structure, but if you find such collapsed structure of a tubule, that is primarily you can identify it as a vein. Now here in this, these are all RBCs, and this violet color nucleated ring like structure in surrounding that lumen is a endothelial layer. Here distinctly you can see the presence of tunica media, a muscular layer, and this is a tunica adventitia surrounding connective tissue layers. Now, what I told you in previous slide here, in comparatively, you can clearly understood why this is artery and why this is a vein. See, clear, distinct, round structure is maintained with large amount of large amount of this muscles layer, tunica media, and clearly distinct distinctions between the tunica adventitia and media. You will find a inter internal membrane you can see here in between these two layer margin forming layers. So in case of artery you can distinctly see this tunica intima media and adventitia. Now in case of this vein or venules, they are much collapsed. Okay, the lumen is much more collapsed, but overall the lumen size is very large, and you see some blood RBCs accumulated in that. But if you try to find out the tunica adventitia, the outermost layer, it is much more fused with the surrounding. And but the tunica media is clearly distinct, and it is good with a small that smooth muscles layer. And endothelial layer is this internal lining of this lumen. Here, the artery and veins are very easy to make apart. The thickness of wall of vein is much less compared to the lumen, and the lumen is often collapsed as shown here. Again, in this same photograph of a section, color sections, this large amount of empty space lumen and no distinct separation between the media and intima layer, and very prominently seen endothelial layer or tunica intima, but and this adventitia is fused with the surrounding. These are the basic features of a veins to identify in a section. Now this photograph is of a major vein in a body. Here you will able to see a distinctively you can see here three different layer, that is tunica intima, media and adventitia, and a large size lumen, but because of this collapsed structure of lumen you can identify it as a vein. Again, another identification characteristic is you don't find any distinct separation between tunica media and adventitia. Okay. They are very much fused with each other. There is no internal margin lining structure is there, and this internal border can clearly seen lined with the endothelial cells. So this way you can identify veins and arteries in a stain color sections. As we have seen different types of artery, here again the veins are also of a different types, like muscular, elastic, and this venules. But here we'll see this muscular vein. Again, the characteristics of muscular vein is presence of large amount of circular muscles in a tunica medium. Tunica adventitia remain throughout the blood vessels, tunica intima remain same. But one more point you will notice in this muscular vein is a presence of vasa vasorum. Vasa vasorum is what? It is a blood supply to the veins. A small blood capillaries, blood vessels, may be arteries or veins or venules or arterioles, providing blood supply to the large veins and large artery, that is vasovasarium. So here you can see the presence of vasovasarium or small blood vessels in a wall of this large vein, particularly located in a tunica adventitia layer. You will never see this vasovasarium in a this tunica media layer, and the surrounding tunica adventitia is all made up of a collagenous dense connective tissue fibers. Now we'll see here some university exam paper questions generally asked on this topic, overall blood vessel topic. Question is a draw label diagram of TS of artery and TS of vein. In earlier many slides, we have seen a basic diagram, schematic presentation of this artery and vein TS. Then sketch and label TS of an artery. These are the frequently asked questions in a university exam. Mostly they ask about the diagram of artery and vein. No question generally asked under the heading of write short note, so long descriptive question is not there. Except this mention the layers of wall of arteries. With the answer could be easily answered like tunica intima, tunica media, and tunica adventitia. Then another way they ask the same question as name the layers of walls of vein. Here again the answer is the same, three layers. Thereafter, in some previous year university question exam papers of mammalian histology, they ask the same repeated questions like name the layers of wall of vein, then state any two differences between artery and vein, which can be easily uh, given answered when you have seen all these slides, and from that you can answer that questions. Like there is no specific demarcations between the three layers, and here muscular layer is more prevalent in arteries, and endothelial layer has internal margin layers. Then state the structure of capillary wall. In next lecture of a capillary, you will see the detailed structure of capillary wall. Still, I answer this questions answer. The structure of capillary wall is made up of endothelial cells. So single layer endothelial cells are with some podocytes or fibroblasts. Then mention various layers of artery. The same repeated questions, three layers, and name the layers of wall of vein. The same answer is there in case of a vein, tunica, media, interna, and adventitious.